Today, I would like to present to you a Pandor bioprocess solution for the growth of biological improvers for biochar. Before I start with the presentation, I would like to show the relevance of today's presentation in this aspect. In the case study that was being presented, and also in the sixth paragraph of the case, it was being mentioned that research was being performed by Senti and Guenadi to employ three types of bacteria as active ingredients in bioameliorants. Bioameliorants are basically organic metals, or rather, they are mixtures that are used to improve the soil fertility as well as structure. So today, I would like to present to you a Pandolf solution for the growth of these biological improvers for biochar. These three bacteria which are being mentioned, are being highlighted in yellow here in the slide that is being presented. So they are the Pseudomonas fluorescens, Flavobacterium species, as well as P. diminuta. Yeah, so please allow me to introduce today's topic of discussion on the next slide. My objective today is to expose the students to bioprocessing as well as bioreactors in general. So I would speak about some of the key parameters in a bioreactor or fermenter process and what Eppendorf can offer in this area. This is the agenda for today's presentation. Let me first introduce to you about bioprocess. The question is, what is bioprocess? Bioprocess is used for industrial application for biological processes involving living cells or their components to generate desired products and improve human living conditions. So in bioprocess, let's break it up into three main categories. Bioprocess can be used to culture living cells or organisms to produce, firstly, in green, food and feed, which is one of the relevance for the topic today. So food and feed, essentially, we are talking about cells making vitamin, amino acids, or flavoring for our food today, or even lactic acid as well as starter culture. So things like your Yakult in daily life, they are produced by microorganisms. In the recent time, popularity has been seen to be grown in the cultured meat industry. Yeah. In the second category, bioprocess can be used to produce biopharma as well as life sciences. Um, I mean, to produce things like antibodies, vaccines, or hormones, as well as growing cells for cell and gene therapy. Finally, in the third category, bioprocess can be used to produce biofuels and platform chemicals. So things like biopolymers or even uh, um, parts that's been used in cosmetics today. These are some of the type of living cells that could potentially grow in a bioreactor. Things like bacteria, yeast, fungi, insect cells, stem cells, mammalian cells, CAR T cells for therapies, as well as plant cells. Right? So what are bioreactors? I have said this word multiple times. So if you are wondering what is it is actually a vessel where cells could be cultivated to produce biopharmaceuticals. They are also control systems to create optimal and controlled environment for the cells to grow in. And in today's market, the size of bioreactor can range from 15 mil, which is in the laboratory scale, all the way up to manufacturing scale, which is 20,000 liters. That is a very, very high volume. And in that volume, it would be in the stainless steel configuration. Yeah. And in the market, there are actually a few type of bioreactors available. There is the stirred tank version, whereby there will be an impeller stirring within the bioreactor to mix it well with the gassing as well as the nutrient and feed that is being introduced into the bioreactor. There is the rocker kind of version, which is also known as a wave bioreactor, where it rocks kind of left and right to circulate your gases and also cells within the rocker bioreactor. And there is also the airlift version, whereby there is no impeller. So um, the circulation is really dependent on the way the gases actually move up and down within the airlift bioreactor. But in Eppendorf, the type of bioreactor that we do supply is the stirred tank version over here. Yep. So why bioreactor? Why did I introduce this to you? The reason why I introduced the bioreactors to you is because they could potentially be used to grow the three type of microorganism or bacteria that were being mentioned in the paragraph 6 of the case study. So by growing all these bacteria, they could potentially be used as active ingredients in bioameliorants. So that is the relevance that we could provide you in today's presentation. And again, why bioreactor? Bioreactor can be considered when there is a requirement for production of large quantity of product. As you already seen in the case study example in the first slide, there could potentially be more experiments that could be carried out to adjust the concentration of bacterial culture and the ratio of biochar to make it uh, work better. So potentially, if you are looking to increase the quantity of bacterial culture, a bioreactor could be of relevance, and it could potentially help you to generate a higher amount of cells within the bioreactor in comparison to shake flask. It also improves the cultivation efficiency to help you improve the cell growth and then further on improve the yield of the product that's coming out of the cell. So it improves cell growth, product formation, product quality by changing the growth condition. In other words, your process can also be optimized to generate your cells in the best condition for your experimental study. So optimization could be performed in such small scale bioreactor. The working volume for this bioreactor is 60 to 250 ml, and you can test multiple parameters in case you are running some kind of process optimization studies for the cell strain that you are trying to improve. So more into what is a bioreactor. It is more than just a vessel. So if you are not familiar with what a bioreactor is, you just imagine it as a fish tank. So I always like to give this example because just like growing fish in a fish tank, you are instead going to grow cells in a bioreactor. Imagine the fish tank as a bioreactor. The tank itself is the vessel over here. The water in the fish tank is the media that you introduce to your cells. And in this case, the fish would be your cells in the bioreactor. And when they move within the aquarium itself, it produces some kind of stirring or agitation within the aquarium that could be translated to how agitation and stirring is being done in the bioreactor. Similarly, daily we introduce food, we feed the fish daily, we make sure that they have enough food, and daily the fish would also produce some kind of waste into the fish tank. That is the same with cells in the bioreactor. And also fishes, they require oxygen to grow and oxygen and some kind of concentration of carbon dioxide. So in the bioreactor, we could also supply all this gas, oxygen, nitrogen, air, as well as carbon dioxide into the bioreactor in a fixed condition so that it can uh, control the amount of oxygen that is going in to be supplied to the cells that you are growing. Other conditions include the temperature of the water or media, as well as the pH condition that is being supplied to the cells. Now let's move on to the next topic and let's take a look at some key parameters in a bioreactor. So I'll be talking or rather speaking about these 6 parameters, which are the key to control the bioreactor condition. First, it would be to give them a sterile environment. Secondly, it would be biomass concentration to be introduced into the bioreactor. Thirdly, it would be the temperature. The fourth parameter would be the pH. Fifth would be the oxygen concentration. And finally, the agitation. Yep, so over here, this is the BioFlow 120 controller unit, and the controlling unit actually controls the parameter on the bioreactor glass vessel on the left, just for your information. So let's take a look at the first point, which is sterile environment. A sterile environment is very important in a bioreactor because while we are cultivating a certain type of cell, we do not want it to be contaminated with another strain of cells that we do not want in the bioreactor, as this would potentially affect your final cells that you want to take out from the bioreactor or product that you do not want in your bioreactor. So it is important that you sterilize the bioreactor properly. So we would generally that all glass bioreactor to go through proper autoclave cycle, which means that if you have tubings or anything on the head plate of the bioreactor, we need to make sure that there is no gap. And we'll load them in something called the autoclave, if you are familiar. And we'll run the cycle at 121 degrees Celsius for at least 30 minutes and with a minimum pressure of 15 psi. So we will stuck a autoclave tape on the bioreactor as well to ensure that the cycle actually passes and reaches 121 degrees Celsius within the autoclave itself. So that is one of the options to keep a sterile environment for your organism to grow. The other option that other operators might be interested would be to opt for a single-use bioreactor option, which look like that. This is our single-use bioreactor known as the BioBlue bioreactor. It comes X-ray irradiated, but some other variants in the market uses gamma and beta irradiation for sterilization, which means that they will come in a sterile bag that looks exactly like this. And once you open the sterile bag, this bioreactor will be ready to use for operators to use, and they are already sterile, so operators do not have to worry about the sterility of the bioreactor. Some other checks that operators could potentially do is to fill the culture media one day before you actually introduce the strain, bioreactor strain into the culture, just to make sure that the operations are all done in perfect condition before you introduce the cells into the bioreactor. The second key parameter that we are looking at would be the biomass concentration. So in a bioreactor, there is a minimum cell density that we want to achieve just to start the cultures growing within the bioreactor. Believe it or not, there's a requirement for pre-culture to generate enough biomass for growth as well. So you will need to initially grow them in flasks before you introduce them into a bioreactor. The need for optimal cell seeding density in a bioreactor is also important. The guideline is typically 5 to 10% of the total working volume of the bioreactor that you will be running. This is because if you introduce too much organism into the bioreactor, it would more often result in an overgrowth of culture, which means that your cells will actually start competing with the amount of nutrients that you allow them to have in the bioreactor, and the end result is that the culture performance will be reduced because of the competition. And on the other hand, if the seeding density is too low, the cells might struggle to grow because there might not be enough growth factor that are being produced within the media to sustain the cell growth. So there needs to be a balance in terms of the amount of cells that you introduce into the bioreactor for growth. And if you are familiar with this, on the right, this is a cell growth curve of a general cell culture. So after you introduce the cells from flask into a bioreactor, they will generally go through a lag phase where where the cells will start to try to get used to the situation or environment that they are being exposed to in the bioreactor. And in this graph or example, it generally take about an hour plus for the lag phase before they feel like it is comfortable to multiply or double in more numbers. And that would be the exponential phase where the cells start to grow and double rapidly within our within the 2 hour time frame that were given. And once they have reached their optimum growth environment, they will start to reach their stationary phase. In some cases where organisms are producing products, this would be the phase that typically people like to perform their process optimization on so that the cell lines could potentially produce more products during the stationary phase. Then when the cells reaches the stationary phase, shortly after that, the cell growth will start to decline and they will start to go to the death phase mode where they start to die, right? And the third key process parameter in a bioreactor is actually the temperature. Every organism has an optimal growth temperature, just like us human beings. And typically, the growth range in a bioreactor would be between the range of 30 degrees Celsius to 37 degrees Celsius, depending on the process. And usually, when the cell grow, this is more common in microbial application, where there are a lot of metabolic activities happening and exothermic reaction happening in the bioreactor. It will generate a substantial amount of heat within the bioreactor. And if this heat is generated in such a substantial amount and is too high in a short amount of time, it would end up causing the cells to die because the cells will start to feel really hot, just like human being. We can't survive in a long-term hot environment. Some of us might get heat stroke without drinking water, for example. But on the other hand, if the temperature is too low, it will start to slow down the cell growth and metabolism, and they will start to grow really slowly. So in a bioreactor, this temperature can be measured and controlled using something known as a temperature sensor or temperature probe. In short form, it is called a RTD. So in a bioreactor, efficient cooling and heating is actually very critical, and this can be achieved when we introduce the temperature sensor into the bioreactor. So now here are some of the ways where the temperature can be controlled in both in all of the bioreactor variants. So Cyvero Twin and BioFlow 320 is actually the name of the control system that Appendorf provides. In terms of heating and cooling, we have several variations. We have the heat block version, whereby heating and cooling is done by the heat block. And then there is this heat blanketed version for the bioreactor. So it generates heat to heat out the media within the bioreactor. And the cooling is actually being performed using cooling fingers. It's a metal piece that is being introduced into the bioreactor. And cold water will be circulated within the Y piece cooling finger to cool down the temperature within the bioreactor. Then there is a double wall water jacketed version whereby if you take a look at this image properly or clearly, there is actually an inner glass wall and an outer glass wall. And between the inner and outer glass door, there are water circulating in between that. This water temperature can be controlled and it controls the heating and cooling within the bioreactor. So that is the water jacketed version. And finally, we do have the last one, which is the stainless steel dish bottom, whereby the cooling and heating happens within this stainless steel dish bottom plate using water circulation as well. So cooling and heating will happen from the bottom up the bioreactor tank. Okay. Now let's take a look at the fourth key parameter in the bioreactor. That would be the pH. Usually in a bioreactor run, there will be pH drift. This is due to the cells growing in the bioreactor and the generation of metabolite waste. So usually at the start of the experiment, you will see the pH being slightly higher. And as your cells continue to grow, you will see that the pH start declining in the process. So what bioreactor will do is that it will respond to the pH drift by adding either acid or CO2 gas, carbon dioxide gas, to control the pH, or it will add in a base solution to control the pH depending on the application itself. So today we want to focus more on fermentation because the microorganism that we are interested in are the three strains of bacteria that was being mentioned in the case study. So for example, if the pH that we set is pH 7 and that pH go above pH 7, which means that if there's a drift from pH 7 to pH 8, then the bioreactor will activate a pump to introduce the acid into the bioreactor so that the bioreactor pH will go back to pH 7 again. And similarly, it works the same. If the pH drops to pH 6 from pH 7, then the bioreactor will immediately activate a pump to introduce the base solution to bring the pH up to pH 7 from pH 6. The next key parameter in a bioreactor would be the oxygen concentration. So this is a major concern for cultivating aerobic organism because oxygen, as you know, is needed for cell growth and metabolism in the form of dissolved oxygen. That's why in a bioreactor, there is also a sensor known as the dissolved oxygen sensor that could measure the amount of dissolved oxygen in the media and tell you whether the cells is requiring more oxygen gas in this moment. Yeah, so this dissolved oxygen can be affected by many factors. I will just list a few over here. It can be affected by the bubble size. So typically the gases is being introduced into the bioreactor by two methods. One is using the overlay, whereby we spush in gas from the top of the bioreactor. And the other way is to introduce gas via the sparger. So this is a L sparger. So you introduce gas through the L sparger so that the gas have direct contact with the liquid culture over here to ensure that once the gas is being introduced in the liquid, it will be fully dissolved inside and circulated for the cells to grow. Yep. So in terms of oxygen concentration, the bubble size is a critical factor. The type of impeller that is used is a critical factor. The agitation rate that is being controlled is a critical factor. And finally, even the temperature of the media could affect the oxygen concentration within the bioreactor itself. Let's take a look at a basic example. So for fermentation, usually when your microorganisms start to grow, it will demand for more and more oxygen. So essentially what a bioreactor can do to get enough oxygen for the cells to grow, one of the few methods that could happen would be to increase the agitation speed. As your impeller increase the agitation speed to a higher value, the amount of oxygen in the bioreactor would also increase. So essentially, it will keep increasing for the cells while the cells continue to grow more and more in cell number. And the agitation also do come to a rate where it reaches its maximum amount of agitation that it can go, usually 1,000 over value RPM, revolutions per minute. Then in this case, the agitation can no longer provide your microorganism with enough oxygen to grow. So in that case, sometimes airflow could be activated to be introduced into the bioreactor so that cells could get some sort of oxygen from the air gas, from the gas flow. And eventually again, when if your cell growth is they grow so rapidly and so well that they run out of oxygen again. And then in that case, pure oxygen can be then introduced into the bioreactor to allow the cells to continue growing. So these are some of the sort of oxygen control that a bioreactor um, can help microorganisms to grow when you are growing them in the bioreactor. So let's take a look at the agitation. So agitation is essential for homogeneous distribution of the culture, the temperature, the pH value, the nutrients that you give to the culture, as well as the oxygenation of the culture. So you can see that in image A over here, when the impeller agitates, it creates a vortex in the bioreactor here. And this actually happen when there is no baffles within the bioreactor. So in general, what people do is that if you are using a really high agitation rate, they introduce things like the baffle. I'm not sure if you can see the dotted red line over here. There are actually 4 dotted red line over here. So you can see that by introducing all this baffle, it will reduce the chances of vortex and ensure a more homogeneous distribution of all your culture, temperature, pH, nutrient, and oxygen within the bioreactor itself. So that is the usefulness of agitation with baffles within the bioreactor. So in the bioreactor, you want to prevent cell settling. And if the RPM is too high, it increases the vortex and shear stress. That's why we always ask customer to consider the baffle option. And on the other hand, if the agitation is too low, then the mixing would be insufficient. And secondly, in terms of the agitation, we look at the impeller type. Depending on the type of impeller that is being used, it generally has 2 type of circulation within the bioreactor. The axial circulation, which is less shear sensitive, and the radial agitation, which is more shear sensitive. As you can tell from the arrow, how the liquid actually moves when the agitation is being started. Yeah. So to give you some perspective into how it works, you can see that the marine blade is using the actual action within the impeller if it's being agitated. So this gives a more gentler agitation for more shear sensitive cells to grow. And if you take a look at pitch blade, it is an actual flow kind of um, agitation with a little bit of Rushton or radial flow, just like the Rushton impeller. So it is it is more, uh, I would say that marine blade is for cells that are more shear sensitive, followed by the pitch blade, and followed by the Rushton blade. So Rushton blade would be for usually recommended for cells that does not need to they are not as sensitive as those who are shear sensitive, which means that usually this is recommended for cells with bacterial cell wall, and in your case microbial fermentation. This would be the most ideal blade to choose in terms of agitation. And then here I would just like to introduce to you a specialized impeller that we do carry in our portfolio in Eppendorf. This might not be relevant to you in this case study, but it's just for your learning and exposure. So this is a pack bed specialized impeller that could be placed in a glass bioreactor vessel, and you can pack this fibrocell disc. The name of this disc is a fibrocell disc. It's 6 millimeters in size, made of polyester fiber and polypropylene. And basically, it acts as an adherent platform for adherent cells to grow while producing extracellular product. So cells can adhere to the fibrocell disc within the packed bed impeller while producing extracellular product into the supernatant, so that we can always harvest the product in the supernatant while keeping the cells growing within the fibrocell disc. Now, I would like to share with you a little bit about the Eppendorf bioprocess portfolio. So in our portfolio, we do offer instruments from the R&D and scale down model all the way to the pilot and production scale bioreactor. We have the Dustbox Mini Bioreactor Control System version, which has the lowest working volume of 60 ml to 250 ml. And this is a parallel bioreactor system, which means that if we are looking to perform process optimization studies, let's say we do not know what is the best condition that we want to culture for this cell line, or if we have many different cell strain and we want to see which are the best cell strain that we could potentially introduce to our experiment in that case, then this kind of parallel bioreactor is very useful because firstly, it helps operators to save a lot of time and also a lot of cost in the media, as one run can within one run you can run up to 4 parameters and 4 bioreactor, as opposed to, let's say, the bench scale system. Within one run you can only run 1 bioreactor at one time. So that is the good thing about the dust box. And then next in our portfolio, we have the dust-kit bioreactor system. So if you just imagine a larger version of the dust box bioreactor system, that is the dust-kit. It serves a similar function as the dust box, just that the volume is greater. It can go up to 1.8 liters. And in this variant, we can control up to 16 bioreactor using one PC. For dust box, we can control up to 24 bioreactor using one PC. Yep. And in terms of bench scale, we have process development. We usually use them for process development. So usually this is being used when there is already a finalized cell strain or finalized process condition that you feel like using. And you would like to further develop this process in a larger scale bioreactor. Then this bioreactor, which is a Cyvero twin unit, or this bioreactor, which is a BioFlow 320 unit, could be used for such experiment. So for Cyvero twin, one controller can control up to two bioreactor at the same time, or you can even run one bioreactor at a time. For glass vessels, we can run up to 3.8 liter. For single use, fermentation goes up to 3.75 liter. Single-use cell culture goes up to 50 liters. And for this variant, which is the BioFlow 120 and 320, 120 is the basic model and 320 is the advanced model. It can go up to go from 600 ml all the way to 10 liter for the glass variant. And for fermentation single use, it goes up to 3.75 liter. For cell culture single use, it goes up to 50 liters. And then the next stage, if we are looking to scale up to even more larger volume, then we are looking at pilot and production scale. Then we do have this kind of steam in place system whereby the autoclave is inbuilt within the system. So you introduce the steam into the system to clean the system. And we have three variations for this steam in place system. We have the BioFlow 510, 610, and BioFlow Pro. The main difference is the working volume of the tank. And finally, we do have the BioFlow 720 unit. Bio 720 unit is a single-use bioreactor that make use of single-use bag and goes up to 500 liter at the moment. So this bioreactors over here that I circle or corner out in red color in the red box actually support our single-use bio-blue bioreactors. So Eppendorf bioreactors are actually flexible in this way because they are compatible with both glass as well as single-use bioreactor on the same controller. Okay, and then we have steam in place and we have the single-use bags. Now, This is the full portfolio of our BioBlue single-use bioreactor. I could say that Eppendorf is the only vendor in the market that is providing you with a rigid wall single-use bioreactor all the way up to 50 liter on the same bioreactor controller. All the other competitors out there is able to provide the solution for you, but it would be on a different controller and they would be using the bag system instead of the rigid wall bioreactor system that you see here. So these are the cell culture single-use bioreactor vessels. They go from 250 mils to 40 liters. And for the fermentation version, it goes from again 250 mils to 3.75 liters. Then this is the specialized pack bed version, whereby there is fibrocell disc being packed over here. We only have one size for this variant because it's a specialized version. And finally, I would like to conclude today's presentation and also kind of end my presentation today to say that the preparation of biological improvers can be performed in shake flasks as they are important tool to grow cultures in bioprocess, but they are not feasible for scaling up to get enough quantity of the microorganisms. So I understand that the research was being performed in shake flasks as well. That's why I did introduce the bioreactor solution in this case. And secondly, scale up is required for controlled vessels scaling up requires controlled vessels such as bioreactors to ensure consistent cell growth and yield within the bioreactor. So if you compare with working with multiple flasks, you can never be sure that all the different flasks are giving you consistent results that you are looking for. And then thirdly, bioreactor is being often used in bioprocess to create optimal and controlled environment for cells to grow and produce products. Next, key parameters in a bioreactor system are really critical for cell growth and process optimization is required to achieve ideal condition for different cell types and cell lines. Finally, I would like to end this presentation by saying that Eppendorf provide a complete bioprocess portfolio to support your bioprocessing needs from small scale to bench and pilot and production scale. And with that, I thank you for your attention and feel free to let us know if you have any questions. Thank you very much for your attention and I hope to see you soon.